I guess. All right, so today we have with us Dr. Alves presenting. So Dr. Anderson Alves was born in Brazil, where he earned his bachelor in animal science from the State University Vale do Acaral, master's in animal science from Federal University of Ceará, and PhD in animal breeding and genetics from São Paulo State University. He is currently a research associate at the University of Wisconsin-Madison and has served as professor in the Federal Institute Education, Science and Technology of Maranhão until 2021. Dr. Alves is broadly interested in the development and application of efficient statistical methods and machine learning algorithms for solving real-life problems related to the genetics and genomics of complex traits with economic importance in different domestic species such as dairy goats, sheep, beef cattle, and broilers. More recently, he also has been involved in precision livestock farming projects, including the development of high-throughput phenotyping techniques driven with different data sources such as digital image and audio signals. So welcome, Dr. Alves, and I'll pass it over to you now. Thank you.
Thank you. Thank you for having me today. I'm really glad to share some of research I've done during my PhD period and some part of the research that I've been doing here at the UW too. Today is very special because I'm here with uh, some good friends that studied with me at the São Paulo State University. So I'm really glad to see those faces, some of those faces here again. And as a disclaimer, if I say some nonsense here, that's because today is April 1st. So let's get started. And so we talk about complex traits, but why are traits complex? From basic quantitative genetics, we've seen that complex traits are influenced by multiple genes and environmental factors. That's correct. That's the basics. But if we speak with our friends from biology or biochemistry, they will say to us, okay, that's true, but there are so much more happening behind those traits. We have so many metabolic pathways. We have other complex nonlinear interactions. For instance, if we think about growth traits, one thing for growth traits to happen should be the growth hormone signaling pathway, and it involves a lot of genes interacting in a complex way, right? So with that in hand, we should conclude that epistasis must be at least at the gene level pervasive in complex traits. So, and apparently we have empirical evidence that so, because for instance, here in Drosophila quantitative traits, Trudy McKay and her colleagues, they were able to reconstruct epistatic networks for those complex traits in flies, in two different flies population. So the question is, how can we analyze such complex traits? The main method that we can use are those methods that I'm naively summarizing as open box statistical methods, which is most of the methods used in animal breeding theory. Generally, those methods, they are linear and they have nice properties. They have interpretable parameters. We can estimate generic covariance components for those models, which is great, right? But since the advent of high-dimensional genomic data, we've been suffering some challenges on how to interpret those genetic signals estimated from those models, because we have so many parameters and sometimes a limited number of observations. For instance, in Bayesian models, we can never the prior effect always will never vanish when we have such a larger number of parameters to be estimated. So and when we are thinking about dealing with nonlinear effects and episteric interactions, for instance, the problem is much worse because we have so many interactions to be estimated. So that's a complex thing. But good news, there is this influential article from Hill and his colleagues that says, okay, we shouldn't be concerned about nonlinear such as episodic effects, because at the end of the day, since the genes are in low frequency, most of nonlinear effects are turned into additive variance in linear models. So that's the reason we generally do not are concerned about episodic and other nonlinear effects. But that's a bad thing also, because it says that even if we estimate additive variance from our traits, it doesn't mean that the genetic architecture is mostly additive, because again, episteric effects and dominance effects, they can generate additive variance in a variance level, even though they are nonlinear effects at the genic level. So the other kind of methods that we've been using, it's what I'm calling here as black box methods. They are black box methods because they have no interpretable parameters, and we generally have to use we have to define some hyperparameters as well, and we do not need to know any prior information about the nature of the problem. So that's the reason we are calling here as black box, because the true model for the nature of the problem remains inside this black box. So basically, we use the input data for predicting or classifying the observations in a nonlinear fashion. And we have a plethora of methods for doing that in machine learning theory. We have regression trees, random forests, gradient boosting, kernel-based methods, and neural networks, for instance, just to name a few. And the bad thing is that we generally cannot interpret the model results in terms of SNP effects or variance components, but we can use those black box methods for a more flexible to gain a more flexibility for a prediction or classification tests, for instance. So here's the question: even though these models, they are nonlinear, can machine learning really capture non-additive effects? So it's, as I'm saying, it's hard to know exactly which effects are being captured within black box method, but we can do some simulation studies to elucidate this question. And this was my first task during my PhD. I simulated dominance effects, and the question was, can machine learning capture some dominance effects without any explicit parameterization? In other words, we are not constructing here dominance kernel, for instance, for the artificial neural network or for the support vector machine. We are using just the raw data, and we are seeing if those methods can capture dominance effects. And the answer was, well, from the machine learning methods tested here, just the random forest was capable of capturing the dominance effects here. As we can see, we simulated two kind of traits with two different narrow space heritabilities, and we were increasing the degree of dominance here for each trait. And as far as we are increasing the dominance degree, the prediction accuracy for the phenotypes in the case of the random forest was increasing as well in both scenarios here. We also tested GBLUP without dominance parameterization and a GBLUP considering also the dominance effects. And the only methods capable of capturing the dominance effects were random forest here and the GBLUP considering the dominance effects. But this model, we have to compute two relationship matrix. We have to add an extra parameterization for capturing the dominance effect. And in this case, we don't have to do any additional step. And when we compared the predictions of random forest compared to the GBLUP methods, we've seen that the correlations are basically higher when we are comparing the random forest predictions with the total genotypic values. So basically what the random forest is capturing is the total genotypic value. And the correlation was even better than the GBLUP-D in this case. Another question that we answered in this paper was, okay, but which effects are being captured? The random forest has important feature that allow us to rank the explanatory variables according to their predictive importance. So we plot here in this graph the predictive importance of each SNP, and we can see clearly here that the peaks for the predictive importance within the random forest ensemble, they are the same as the KTL, the simulated KTL locations, especially the additive effects here. But there are some other peaks that are also capturing dominance KTL solely. So that makes sense because this trait here, the simulated trait had a narrow sensitivity of 0.30 and the dominance degree was 0.50. So the additive effects should be more prominent than the dominance effects, but still the model was capable of pointing for some dominance KTL here. I didn't investigate the epistasis effect during my PhD, but fortunately there are so many good papers that did that on the literature. Here we have some example. In this case, they simulated a lot of complex traits with different generic architectures, and the results were here in purple. We can see a complex traits that were influenced by epistatic effects, and they showed that the machine learning methods, they were better than the parametric models in this case. When we have epistatic effects, some methods such as random forest, support vector machine, and Gaussian process, they overcome the prediction accuracy of parametric models solely. But why? Why is that? Why can machine learning methods potentially capture epistasis even if we do not add any extra parameterization? Well, for answering that, we can recall here the classical epistasis model here for two locus. Here we have the Y observation. We have here the X1 locus, X2 locus, and the interactions. And if we take the derivative of the Y observation here regarding to the first locus, we can see that the effect of the locus 1 on the y doesn't depend solely on this marker, but depends also of the levels of locus 2. Here, this is what the derivative is telling us. We can do the same for instance, for kernel-based regression. Here we are using Gaussian kernel. And if we take the derivative of this model, of this observation here, for the animal I, regarding to the marker genotypes for this animal, we can see here, if I'm doing the math correctly, that the variation on the animal I does not depend solely on this animal genotype. So this is a kind of epistasis not represented by linear model, because this model is linear regarding to the parameters, but it's non-linear regarding here to the input data. And we can do the same for instance, for tree-based methods such as random forest and gradient boosting machines. These methods, they are based on what we call regression trees or classification trees. They build a lot of regression trees, and then they average the predictions within those regression trees. Here I'm building just one example of what should be a regression tree. And this tree is built based on a splitting process of the SNPs. And the prediction at the terminal nodes here, they are done by averaging the subspace of the observations here. But we can see here clearly that this regression tree is telling us that the effect of SNP1 on the trait depends also on the levels of the SNP2 and SNP4 in this case, in this branch here. And the effect of SNP1 in this branch depends on the levels of SNP3, SNP7, and SNP9, and so on. So this is also a kind of episteric interaction too. Knowing that we applied machine learning for real-world data, in this specific case here, we applied machine learning for predicting reproductive traits in a lorry cattle, using both parametric models and machine learning methods. And we used for that about 2,000 to 5,000 animals, genotype animals, for about 300,000 SNPs. And here's just to exemplify why it's complex to sometimes use machine learning methods for predicting. Here we've tested different hyperparameters for the support vector regression, and we can see here clearly that the hyperparameter that we choose, it matters for the prediction accuracy. And this depends on the level, depends on the trait as well. So we must fine-tuning the correct hyperparameter for each support vector regression here. And the same was true in the last stint for the artificial neural networks. The number of neurons tested made some difference on the prediction accuracy, that depending on the trait. In general, the support vector regression overcome the parametric models in this case for all traits, even though some traits were expected to be affected by linear, by additive effects. On the other hand, the random forest and the artificial neural networks, they didn't overcome the parametric models in this case. But I would like to highlight that this is just a specific result. If we search in the literature, we will find that sometimes machine learning can overcome or cannot overcome parametric models. It will depend on the traits and on the population that we are working with. My third task during my PhD was, okay, machine learning are black box methods, but can we use some of them to learn something about the generic architecture of the traits that we are working with? So we did this genome-wide association study, but using a random forest approach. And it turns out that we found a lot of genes related with fertility for this specific trait, which was age at first Kelvin in a lorry cattle. So what we did, we first ran 5 independent runs with different seeds for the random forest. So we've run five independent random forest analysis, and then we computed this F score. So the higher the F score, the higher the predictive importance for the SNP, right? And following some suggestions, we've set a threshold of 3 for identifying relevant SNPs on our genome-wide association. After identifying the relevant SNPs, we search on the database for candidate genes for those SNPs. And we also did a functional analysis using cytoscape for the candidate genes. Since the F score here that we are computing doesn't tell anything about the effect that the marker is capturing, we did some additional analysis with a mixed model approach. We took the SNPs flagged with the random forest analysis, and we analyzed them in 2 ways. The first way was in a single marker model using each SNP as an additive way and correcting for the population structure here. The second model was testing for every pairized episteric interaction between those SNPs. So we got the relevant SNPs and we tested the 2 main effects plus the interaction effect here, and also again correcting for the population structure by using a genome relationship matrix. So here, as a result, we can see that for all five random forest independent analysis, they were pretty stable. They identified the same genomic regions, and then we averaged the F scores for all those 5 independent runs, and we used the average as the final importance score for each SNP. So doing that, we found 117 SNPs associated with age at first Kelvin. Those were the SNPs surpassing the threshold of 3 here. And surprisingly, most of those SNPs were at exon or intron regions of candidate genes that have known roles for fertility-related biological functions. And we could see in every peak here that we can find a fertility-related candidate gene. After that, we did a functional analysis, and it was confirmed. For instance, here we can see that at least 11 genes are associated with embryo development, which is clearly associated with age at first Kelvin. Another cluster of SNPs were associated, were enriched for pheromone receptor activity, which also has something to do with fertility traits, right? And a huge group of candidate genes here were associated with growth factor stimulus as well. But the second question was, these effects that we are capturing, are they additive or are they aesthetic? So we used the mixed model approach for answering that, and we could see that the correlation between the p-values and in the mixed model approach, considering only the single marker association, and the f-score in the random forest was just 0.6. So there is, they have a high correlation, but they are not agreeing thoroughly, right? So that's why we can see here on the diagonal here we are seeing the p-values for the single marker effects, the analysis that assumes that the markers had only additive effects, and we can see that hopefully half of the SNPs were non-significant for additive effects. But these same SNPs, they were involved in episteric hotspots, significant episteric hotspots here. For instance, we can see that a lot of SNPs in the chromosome 11, they present significant interaction with SNPs in the chromosome 3. And some SNPs on the chromosome 21, they are interacting with SNPs on chromosome 5. And here we have a huge interaction of chromosomes 11 and 24 as well. So we found about 700 episteric interactions with nominal p-values less than 5%, which gave us a rejection rate of about 11%, which is twice that expected just by chance, right? And correcting for the false discovery rate at 10 and 5%, we end up with 7, 5, and 6, 5, and 7 SNPs respectively. And those SNPs were located at chromosome 3, 5, 11, 18, 21, and 24. Here we are just showing some of these SNPs that had significance episteric effect, and we can see that a lot of interesting results here. For instance, a SNP in the intron region of this gene is interacting with a bunch of genes in the chromosome 3 here, at least 4 genes, right? And we also can see that there is a hotspot of interactions between these genes, genes here at the chromosome 21 with this unknown gene at the chromosome 5, right? So, and it should be, we must highlight that those SNPs here wouldn't be identified by parametric model assuming additive effect. So that's this highlights the importance of using different methods for knowing as much as we can about the generic architecture of a trait, right? Another thing that we are doing here at University of Wisconsin, it's working with the idea of multi-task learning for genomic prediction. We are investigating if we can do multiple trait genomic prediction with nonlinear, non-parametric models. And we've been working with some multi-task kernels for doing so. And we've been developing some of those kernels for plugging into the support vector regression for multiple traits. And we've been working with this Q kernel here that has 2 plus t square hyperparameters that we must decide the best value for those hyperparameters. And that's a problem, because if we think about only 2 traits and we are supposed to test at least 10 values for each hyperparameter, we would end up with 10 to the power of 6 combinations in the hyperparameter space. So that's huge, and we cannot perform a traditional grid search for doing so. So the idea was to use a generic algorithm for fine-tuning the model hyperparameters in this multi-trait support vector regression. So the idea was to represent the hyperparameter combination within those binary arrays here, and each array give us a prediction accuracy, a prediction error that we are comparing as a fitness function. So basically, we generate an initial population of models. We assess the best models on generation in n-1. We select the best models, individuals, or wherever, and we create a new population based on the best individuals of the previous generation by using techniques such as crossover and mutation. So we create this new population. We assess the fitness score again, and we do this process over and over again until we find the best hyperparameter combination. So sounds scary, but we've worked in a first step to program this concept in our package. So the first step was done, and we can right now to implement this just in a few lines of our code, which is awesome. And another interesting feature is that we can do the generic algorithm optimization in a parallelized process. So we can boost the computational time here. In this case, we are using 7 cores in this parallel processing. So we tested our new method for predicting Carcass traits in broilers. We are working with a pure breed line that was bred in 20 generations. And we are working here with 2 Carcass traits and also with 2 in vivo traits that we are calling growth trait and feed efficiency trait. Here I'm not speaking the true name of the traits because of course the data is commercially sensitive. But we are working here just with animals with high EBV accuracy, which gave us about 5,000 genotyped individuals. So what we did, we split the animals according to the generations, and at the first stage, we used the animals from generation 1 to generation 12 for training the generic algorithm for finding the best hyperparameter combination. And the generic algorithm assessed the values on the generation 13 to generation 50 here. And the target population was unavailable at this time. So we first found the hyperparameter combinations using the generic algorithm. Then we retrained the models with the best hyperparameter combinations using the whole training data in this case. And in this second stage, the aim was to predict the information of the target population for the Carcass traits. And we did that using three different training strategies. First of all, we trained only single traits models using training from generation 1 to generation 15 and predicting from generation 16 to generation 20. The second strategy was using a B trait model. Again, here the target trait was the Carcass trait, but we are using a secondary trait for data augmentation here. But for this second strategy, we are not using the information of the target population as well for training the models. And finally, the third strategy was using the whole data set for the secondary trait, which should be growth or feed efficiency, for predicting the target information here, which was the Carcass trait 1 or Carcass trait 2. And in this case, we are just using the information of the primary trait from generation 1 to generation 15. So just to summarize, the traits analyzed were in single traits models were the Carcass trait 1 and Carcass trait 2. For the B trait models, we analyze Carcass trait 1 with growth trait and Carcass trait 2 with feed efficiency trait. We compared single trait GBLUP, multi-trait GBLUP, and the proposed methods, single trait support vector machine, a single trait support vector machine weighted for some genetic architecture that we computed the weights by doing a genome-wide association on the training population. Here, this model is the multi-trait support vector regression without waiting for the genetic architecture. And in this case, we have the multi-trait support vector machine, but waiting for the generic architecture. So the GBLUP and multi-trait GBLUP were fitted using a Bayesian inference using the BGLR or MTMT package, and the support vector regressions optimized with the genetic algorithm were fitted using our under development package written in the R environment. So here, just to finish up, we have some preliminary results. We can see that the generic algorithm is learning something about what is the best hyperparameter combination, and this learning process was best until the generation 10 of the algorithm, and after that we kind of get stuck in a local minima. But yeah, it answered that we are learning something about what is the best hyperparameter combination for those models. And in this figure here, we have some predictionability results. And the best method here was the proposed method. For instance, for Carcass trait 1, we achieved a prediction accuracy of 0.79 for the multi-trait support vector regression using the third training strategy, whereas the performance of the multi-trait GBLUP was about 0.72 here, as we can see. So our model clearly overcome the multi-trait G-block. And the same happened for the Carcass trait 2. We achieved an accuracy of 0.8 for the multi-trait support vector regression using the third training strategy, whereas the performance of the multi-trait G-block using the same training strategy was 0.76. So we can see clearly that we can gain an extra prediction accuracy by using those nonlinear support vector regressions models. Unfortunately, the accounting for the generic architecture didn't improve further our model performance here, as we can see here in this last bar. Okay, so I think my time is almost there. So just to finish up, we also did some comparison regarding the time for running those models. And the single trait and multi-trait G-block models, they took about 1 hour and 300 minutes each for running the Gibbs sampling. And our proposed method, they took about 30 minutes for the single trait case and about 7 minutes for the B trait case. So this was the time for building the block kernel matrix, compute the Euclidean distance, build the block kernel matrix, and to run the generic algorithm for 30 generation. So again, we must highlight that the process was done using a parallelized process. Okay, so just to finishing, we've seen that machine learning methods, they can be attractive for flexible genome-enabled prediction. Some machine learning, they may overcome some challenge that we are facing with post-genomic data era. For instance, these days, researchers, they are interested on combining multi-source data from multi-omics data for prediction. And this could be challenging using parametric models and could be easier using machine learning methods, for instance. Also, some methods, they are not completely black box. We can learn something about the generic architecture of the complex traits, for instance, by using assemble learning methods such as random forest or gradient boosting machine. And finally, the multitask learning is a potential approach for dealing with the high number of novel traits that are being generated with high throughput phenotyping technologies. So in this case, we have many traits to analyze, but we have a limited number of observations. And that's a complex task to estimate covariance components in parametric models in this case. And we can overcome this limitation by applying multitask learning techniques. So I would like to thank the São Paulo State University, where I did my PhD. Also, the São Paulo State funding for financially supporting this work. Also, the GeneSys for providing the Nelore cattle data. And of course, the University of Wisconsin for hosting me as a postdoc in this new stage of my life. And also the COB Ventures for providing the data and for providing also financial support for this work. So thank you very much. I think I'm done.
Thank you, Dr. Alves. That was a very good presentation, full of information, very new technologies right there, right? So now for you in the audience, if you have any question or a comment to Dr. Alves, you may raise your hand. There's a reaction here on Teams that you can do it. So, or you can also use the chat box to ask him a question or make a comment. And you also may open your webcam and unmute yourself for that.
Yeah, should I, just a question, should I stop sharing my screen, because right now I cannot see you, so.
Okay, well, yes, then you can stop sharing, and if it's necessary, if someone has any question related to any slide, you can go back to it.
Yeah, just a second.
Yeah, no problem. Can you see me now?
Yeah, I'm seeing you now.
Awesome.
Let me check.
So, yeah, I see Lucas Lopez has a question. So, Lucas, please go ahead.
Anderson, good to see you, man. It's been a while.
Yeah.
Glad to see that you and Rebecca are doing great there in Wisconsin. Quick question, I might have missed it. How much data did you have for this for the studies that you had? Because I know that like applying machine learning is we usually run into the problems of having like not enough data for basically everything. So how much did you have, and how did you handle that?
Yeah, it was relatively a small data set. For the Nelore Carroll data set, we had about 3,000 to 4,000 genotype animals for about 300,000 SNPs. So that's our data. So it's not a small data set, but it's not a huge data set. And for the broilers data set, we had 5,000 animals genotyped for 6,000 SNPs. So they were basically the same size.
Okay. Yeah, no, it's a reasonable data set.
Yeah, it's a reasonable data set. Yeah, for sure.
Yeah. No, again, just great to see you. Great to see that like things that we saw starting back in 2018, like now you're presenting here. It's just awesome. So
yeah,
congratulations.
That's crazy. Yeah, thank you, man. Good to see you too.
Lucas, you still have a question? Or okay. So does anyone have a question?
Just lagging a little bit.
Okay. To Dr. Alves. So I think I have a comment that is a comment slash, oh, I see Flavio. Flavio, do you have a question to Dr. Alves?
Okay, I was trying to open my camera here. So, I have a question for Anderson, and maybe a comment, right? Because I think you clearly show that we can increase, we can predict better non-additive effects, so the genotypic effect of the animal. So, and that would reflect in broad sense heritability for the trait, right? But because if you use like these machine learning techniques, what you are predicting. You don't know what genetically you are predicting. You know that you can predict back the whole genotypic effect.
Yeah.
But part of the you are predicting now is not going to be transmitted, right? For sake of selection. Only if you if you're working like a species like swine or poultry, that you are working with cross, you like to predict the outcome of the cross, right?
Yeah.
To exploit this let's say heterosis. So have you looked at Anderson in a way that if you take into account the non-additive effects, if you could also estimate better the breeding values based just on the additive effects, like the allele substitution effect. So I think that's is important, right, for selection. If you take into account in the model like dominance, if you can predict better the breeding value that just based on allele substitution effect. Have you looked at this?
Yeah, actually, for this last example that I gave you guys, actually the target variables, they were the breeding values. So that's a thing. Even though the breeding values, they are supposed to be additive, we have some epistasis on the genic level, right? Even though this epistasis just doesn't generate a substantial amount of variance, we could increase the prediction accuracy for breeding value as well, because uh, because epistatic effects, they can generate additive variance,
Yeah.
and also because we have the opposite as well. We've seen that linear systems, they can generate false epistatic effects as well. So yeah, for this last case, we used the breeding values.
Oh, okay, so I missed that. So you calculate all those accuracies against the breeding values, right?
Yeah, I was running out of time and I didn't make it clear, but we used only the animals with high breeding value accuracy.
Yeah, so you kind of target was the breeding values, right? You are
The
trying
target was the breeding
to predict.
Value.
Okay, that makes a lot of sense now for me. Yeah.
Thank
okay, good that I asked this question because I didn't.
No, it was totally my fault because I was running out of time and didn't explain it better.
Okay. Thanks a lot, Anderson. Very nice presentation.
Thank you very much.
Congratulations.
Nice to meet you, Flavio.
Yeah, my pleasure.
Yeah, Dr. Tupan, if you want, please go ahead.
Thank you very much, Ivan. Thank you very much, Dr. Alves, for an awesome presentation. And sorry for my voice, I'm fighting something here, but anyhow, not important. What's important, I have a follow-up question based on Flavio's question. And, you mentioned that you use breeding values as guidance for the optimizing, basically for prediction. Typically, when people use machine learning, you need to have observable values as the target. Breeding values, to my understanding, they are computed values. These are also predictions. So if you use them as your guidance to match with predictions from a machine learning, then you basically pit your machine learning model or algorithms that create the model against whoever predicted or whatever predicted those breeding values. Is there a so-called ground truth breeding value out there, like a true breeding value? Based on what I know, I don't think there is, but I don't know. Maybe I don't know. Thanks.
Thanks for the question. That's excellent question. And what happens that, okay, breeding values, they are computed values. They are smoothed by some linear function, but they require a lot of information for getting a reliable breeding value, right? So the thing is to use the data that we've that we have so far for building those high reliable prediction, high reliable breeding values, and use those highly reliable breeding values for training a machine learning, so that in this case we are predicting the breeding values for future generations, but just using the genomic information, right? So in this case, we don't have to use a lot of phenotype information, and this makes sense in this case because we are working with Carcass traits, which are hard to measure, which are expensive to measure. So we don't have so much phenotype information for estimating good breeding values. So we are using those those breeding values with high accuracy for training a machine learning method to predict the future generation, right? I don't know if I answered that. So, and the thing is, we can use the breeding values of easily measured traits for boosting the prediction accuracy of the prediction for traits that are hard or expensive to measure, such as Carcass traits. So in my opinion, yeah, we can, of course, we have to apply a linear method first to estimate good breeding values. But the amount of breeding values with high accuracy, it's limited by the information that we have. So we can use machine learning methods for using what we have so far for predicting the breeding values of the new generation, right?
Thank you for the clarification. I mean, I have probably a different opinion or a different. I would have tried something else on top of this. I think it's okay to use it as a guideline, so basically to direct a little bit the direction. But I have a feeling in my head also some unnecessary bias at some point, depending on the data that you use, how much you have, how much is missing, how well it samples the whole population, etc., etc. Yeah. So without going into details, I wonder, what do you think about, instead of using a breeding value as the guidance or the target to predict, why don't you try to predict the traits directly?
Yeah, we can do that as well. But that's the tricky part, because since we cannot do some inference about the machine learning predictions, we should assume that we are predicting the target variable, right? So if we are training a machine learning with the phenotype, we should interpret is a prediction for the phenotype. We cannot interpret this as a prediction for the breeding value itself. So if you want to predict breeding value, we should training our machine learning method with breeding value. So it's a matter of the scale of the trait, right? But yeah, for sure, we can use the phenotypes for training a machine learning method in the case that we would like to predict the real phenotype, and so that we could take some decisions based on those predictions. So that's totally possible.
Absolutely. Sorry, I probably omitted the direction of the goal of the study, right? So if your goal is to actually align with breeding values, absolutely, that's no problem. I don't know. I can ask one more question. I promise it's the last one.
That's for sure.
I'll try to ask it quickly. Probably it wasn't in the slides. I'm sure it's probably somewhere in the papers, which shame on me, I didn't have a chance to read all of them, just one. I'm working on it, by the way. I wonder if, sorry about this. I wonder if there is any particular part of your study where you actually looked at, basically tested the overfitting of the models. Random forest and particularly the decision tree-based methods, regardless of how you combine them, they are really famous for overfitting terribly. Random forest, not so much, because it has inner mechanisms to do it, but assuming that they don't, that's totally wrong. You need to check, you need to test. Did you guys look at that, or is it in the plan to look at it?
Yeah, For prediction, we did a cross-validation to make sure that we are not overfitting the data, right? And for the genome-wide association study, what we did was to try a lot of different hyperparameters and to look at the out-of-bag prediction error, which is not 100% accurate, but it's a guideline for how good the model is feeding the data, right? And of course, the main empirical evidence was the biological function of the genes identified with the random forest. All of them, basically, all genomic regions, they were related to fertility functions, which is awesome, which support our findings.
Okay, excellent. Thank you very much. I promise last question.
Thank you.
Yeah, thank you, Dr. Tufan, for good conversation, good discussion here, actually. And I think there are some messages in the chat, Dr. Alves, I think you're able to see it. They're not actually questions,
Let
just
me
some
look into
comments.
It. All right. There are some comments.
Yeah, they are basically comments. I was just make sure that you could see them.
Thank you all for the comments.
Yeah, and I think we don't have any more questions. So once again, thank you so much, Dr. Alves, for accepting our invitation and for giving this.
It was my pleasure.
Yeah, very good. And thank you all for attending the seminar today. Once again, as a reminder, for next week, we'll host 2 CGIL seminars, one on Tuesday at 3 p.m., and it will be a hybrid format, so there will be we will stream the CGIL seminar as well as the one on Friday. So don't miss that. And I hope to see you all next week.